All right, good afternoon and welcome back to FirstThought Focus. FirstThought is a life sciences expert network with the mission to bring subject matter expertise back into the expert network industry. FirstThought Focus is our virtual conference series that aims to showcase the breadth and depth of FirstThought's experts, while also covering a wide range of topics that are relevant and important to the life sciences space. Last month, we wrapped up a 4-part series on CAR T and cell therapies and a 2-part series on the human gut microbiome. You can find the replays to these events on our website, www.firstthought.io/focus. Today, we are bringing you the first in a series of events on gene therapies with Dr. Gao from the University of Massachusetts Medical School. You will also be able to access a recording of today's call on our website as well. And just finally, as a quick reminder before we get started, we will be taking questions from those of you who are listening in live. You can submit your questions via email to hello@firstthought.io. Again, that's HELLO@firstthought.io. And with that, I'd like to turn things over to our moderator and veteran science journalist, Neil Canavan.
Welcome to a FirstThought talk on the origins, anatomy, uses, and related challenges to the commercialization of AAV gene therapy. My name is Neil Canavan. I'm the managing director of the KOL network here at LifeSci, and I'm a host for today's webcast. I'm also the author of A Cure Within, Scientists Unleashing the Immune System to Kill Cancer, which you can get a free signed copy of if you just send your snail mail to Morgen. Now, the reason I brought this up is my next book is on gene therapy. And the subject of one of the chapters in this book is Dr. Guangping Gao, and he is my guest today. Dr. Gao is a professor of microbiology, physiological systems, and biomedical topics, as well as the director for the Gene Therapy Center and Vector Core at the University of Massachusetts Medical School. Pertinent to our discussion today, 2 things. Dr. Gao is not merely an expert on AAV. He, in fact, discovered many of the AAVs now used today in gene therapy. Further, and I can't resist mentioning this, Dr. Gao also discovered the gene for Canavans disease. Now, I have no relations to the person who actually named this disorder, but there it is. It's a small world. Dr. Gao, welcome to our webcast today.
Thank you, Neil, for the very nice general compliment in introduction. Thank you.
Okay, so as luck would have it, when FirstThought first invited you to speak today, you shared with us a brief introductory slide that you recently put together illustrating the main issues in the field. If you would be so kind, I'd like to start with that introductory slide today. And if you could share your screen and tell us about it.
Yes, I will be more than happy to do so. Hopefully we can make this work. But this is often, yes. So the slides Neil was talking about is a slide I made to kind of summarize in the adventure for commercialization and translation of the gene therapy from proof of concept to bedside. One of the major barriers, personally, I believe, in our roads, gene therapy road 66. I believe the first one is how to make large quantity and high potency vectors at a reasonable cost, because that would become access issue for gene therapy, affordable issue for gene therapy for patients. So that's the first barrier when you get out of Chicago on the road 66 of the gene therapy. When our Gene Therapy Express continue to move on these roads, we're going to run into second challenge or barrier. That is pre-existing innate capsid and transgene immunity. And that is something we got to overcome if we want to deliver gene therapy from bench side to bedside. The third issue is we need to have better delivery vehicle, better AAVs that is desired tissue tropism, cell type and tissue type specificity and efficiency to achieve safe and effective gene therapy outcome. Finally, right now we're talking about gene therapy. We thought the more the gene, the better. But actually soon we will realize that we got to consider regulated gene therapy, particularly if we can regulate it in a physiological way. That's the best gene therapy. So once we overcome all those barriers, then we should be able to deliver gene therapy to the bedside to benefit all the patients and save their life or improve the quality of their life. Thank you.
Dr. Gao, thank you for that introduction. And a quick technical note to our listeners, no, you are not having a stroke. We actually have a little bit of streaming problems on Dr. Gao's end. So if you could be patient with the audio on that. So let's get started now that we have the roadmap. I'd like to warm up the engine first with some more basic science. Let's start with naming. AAV stands for Adeno-Associated virus. Dr. Gao, the name implies that adenoviruses, like the kind that cause the common cold, are somehow had something to do with AAV. Could you tell us briefly how AAV was discovered?
Yeah, AAV was discovered back in early 1960 or middle 1960. And that was actually discovered as a contaminant of a common cold virus called adenovirus. And AAV actually to date, one of the major attractiveness and advantage with AAV-based vector is AAV so far has not been associated with any human disease. So that was discovered, as I said, more than half a century ago. And at this point, I think the AAV has rapidly developed into a gene delivery vehicle for gene therapy.
Okay, well, let's go ahead and have a look at it. Let me see. Well, there it is. Gosh, it's not really much to look at. It's a beach ball, it has DNA inside. The ball is only about 22 nanometers in diameter, and the ball is actually called a capsid. The DNA contains a single-stranded and consists of 4.7 kilobases. This is pretty small if you think about other viruses like COVID has like 26 or 31 kb, much larger. But the point I'm trying to make here is how much genetic material of therapeutic interest one can pack in this tiny ball. And we're going to discuss that a bit later. But first, I want to bring up, I want to continue with the basics. And I'll bring up another graphic. And this is a brief walkthrough on how the virus can take over a cell. Dr. Gao, if you could walk us through that.
Yes. So first of all, AAV as a virus, just like any other viruses, in order to get into cells, they need to somehow bind with receptors on the cell surface. And then it gets through an internalization process, get into the cells. Just like many other viruses, AAV has to go through so-called intracellular trafficking. Those steps include, first, get it through early endosome, that one of our cellular organelle in our cells, and through some pH changes, the AAV escaped from endosome and then enter the nucleus where we have our genetic material. And there, AAV uncoat or take up its capsid and expose its AAV genome. In that case, AAV, in most cases, is a single-stranded genome that cannot produce any gene products. AAV has to go through that process, so-called either second-strand synthesis or self-annealing, because AAV is packaged as positive strand and negative strand, and they can kneel together. In both mechanisms, AAV become double-stranded. That can produce the gene product we need. However, that's not enough. AAV figure out a mechanism to stabilize itself. The predominant mechanism is called circularization or concatenation. they just join their ends and form circular molecule or concatemer molecule. Very, very small portion of AAV may get integrated into our host genome, but that is very minor event. That's the process. Once we have that integrated, once we have that stabilized as a circular genome, once it becomes a double-stranded, AAV start produce its therapeutic gene where if it's Y-type viral gene.
Okay, I want to talk about how the immune system does not recognize this very foreign circle of stuff that's floating around in the nucleus. Strikes me as rather remarkable. But there's another virus that just for context, lentivirus does actually insert into the home genome, does it not? We're also using that in some gene therapies.
Correct. So in the gene therapy, there are two major approaches. One called in vivo gene therapy. AAV is the most attractive tool for in vivo gene therapy, meaning that you deliver gene therapy drug directly into human body and achieve therapeutic outcome. Another virus is another approach so-called ex vivo. In this case, you use virus like a lentivirus. You take out human cells, you manipulate or genetic engineering, use lentivirus in cell culture, and then you modify the genetic modified cells, you put back to human, and then that modified cells will function as a living drug in human body, accomplish therapeutic outcome. But that requires integration into the cells you modified.
Okay, so lenti integrates, AAV usually does not.
Correct.
Does that say anything about the persistence of the transfection? Is one more stable?
That is a great question, because for lentivirus, because it integrates into the cells you modified, but for AAV, if your target cells, like mitotic cells, start division, cell division, and one cell become two become four, and blah, blah, keep going. By doing so, then you will gradually lose your vector, your vector genome will get lost. However, if AAV can transduce, we know AAV, a preferred target for AAV gene therapy would be those terminally differentiated cells, non-mitotic cells. There, AAV, because it's circulate or concatenarized genome, and then AAV can stable in those cells, accomplish long-term therapeutic effect without cell division. And cells tissues include CNS, eyes, muscle, liver. Those are the good examples and good targets of AAV in vivo gene therapy.
I'm going to come back to the target tissues in a little bit and reiterate some of what you just said. But first, I want to stick more with the basic science of how to do what we're doing here. As with all science, this all looks really easy on paper, but it's kind of a pain in clinical practice. So let's look at the challenges to translation. One of the biggest issues is toxicity, or the clearance of the AAV vector due to immunogenicity. So you lost it, nothing works. So Dr. Gao, the biggest problem here is pre-exposure to AAV. These things are really common. Almost everyone on Earth has seen one type or another. And that means the immune system recognizes it as foreign, saw it before. It has antibodies ready to go to attack it and neutralize it. And well, this is either toxic or you've lost your graft. So what are some of the mechanisms in place that are a threat to what we're trying to do here?
Yes. So the mechanisms are the following. First, we call pre-existing immunity. And in that case, you can see a virus was binding to an antibody called the neutralization antibody. That is because, as you said, a patient may have previous natural infection, generate antibody against a specific cell type of virus. When you have the gene therapy vector get into your bloodstream, then those pre-existing neutralizing antibody will find those viruses and bind to it, neutralize it, and cancel its gene therapy effect. So that's the first step. The second step, AAV combining to some toll-like receptors on the cell surface, and then that could trigger some innate immunity to AAV capsid, AAV variant. That's second immunity you could happen.
If I could interrupt you for just a second, the TLR, in this illustration, TLR2, TLR are a generic thing that recognizes an invasion of any pathogen. It's not, yeah, okay.
It's a pattern recognition molecule. So that is more generic than the AAV specific.
Got it.
And once you get in the cells, then particularly when AAV started exposing its genome, and then you have another line of defense, that is a TR9 or other similar molecules that will recognize DNA sequences in those AAV gene therapy vector genome. And in that case, will trigger DNA sensing and immunological innate immunity. So that's a third layer. And the fourth layer is when we finally, AAV, after pass all those barriers, get into the nucleus and start express its therapeutic genes, then you have two more potential immunological barriers. One barrier is that your protein or transgene products will start get recognized by the body by the B cell or we call humoral immunity and generate antibody against your therapeutic gene product. The second pathway is those transgene products will be processed by our very strong immune system and very stringent immune system will present those therapeutic gene products on the antigen presenting cells. And then those presented antigen will be recognized by T cell through T cell receptor. And then we generate the T cell or cellular response against either capsid or our transgene product. So all those collectively kind of our body build up a complete defense system against the gene therapy. That's what we have to overcome as a gene therapist.
Gosh, it all sounds incredibly simple. I can't imagine why we don't have gene therapy today. So let's talk about what to do about it. We got a lot of problems here. Let's think about what kind of solutions we might have. I suppose in the last example, one drastic solution to eliminate an immune response is you could deplete the body's, an individual's B cells, which that's what generates the antibodies. If you wipe them out, then you could deliver your vector and that would be just fine. That's pretty drastic. What else can we do? What other technologies are you looking at to avoid this immunogenicity?
Yeah, because I think the key challenges are at 3 different levels. One level is before you give gene therapy to patients, you probably need to figure out which patient, what population of patients that may or may not have a pre-existing immunity. So you can screen their serology against AAV capsid and serotypes, and to select the patient that's immune negative to the virus. So that's one possibility. Second possibility is that you can do things such as plasmapheresis. Then you remove those IgG specifically to those AAV capsids.
Right.
And third strategy, you can do immune suppression to suppress immune response against your gene therapy. And so additional strategy, particularly if you want, so right this moment, our treatment rely on AAV is persistent. One shot work for at least a decade. But if we need to redose the patients at some point of your life, then we have to think about what we talk about immune suppression or other method we just mentioned, but we also could evolve our capsid by different ways to make it more kind of escape from our immune systems. So those are the strategies we can take, we can do to overcome this immunological barrier.
Okay.
Barriers.
But again, I mean, getting rid of all your antibodies or using rapamycin, there's some difficult prospects here. For the things that you've just listed, what are you most hopeful about? What do you think is the best way to go?
So at this point, I think one of the major directions for the field is capsid evolution. So people trying to use artificial intelligence or rational design or natural or direct evolution to generate some capsid can escape from pre-existing immunity and then can dose the patient. So that is one of the very important steps or approaches to overcome these immunological barriers.
Okay, I see. Now, before I ask my next question, I'd like to mention that the last two images were taken from Dr. Gao's excellent review paper in Nature Review Drug Discovery. This is in 2019. This is titled Adeno-Associated Virus Vector as a Platform for Gene Therapy Delivery. It's an exhaustive review. It's really quite good. Now, to be clear on this issue of immunogenicity, in gene therapy, a really strong immune response can kill you. And this has recently occurred with the therapy developed by a company called Adeno-Associated using AAV8 to treat a neuromuscular disorder. Dr. Gao, in this specific instance, is it known what caused the reaction? Is it the capsid, the transgene, the number of vector? Does anyone know yet?
That is a great question. First, as a gene therapy researcher, I feel very sorry for those patients. And so, but same time, this mechanism or a toxic mechanism still unknown. I believe the company is still doing a lot of research to figure out. But personally, I believe is the following possibility. The first, I would say, is underlying disease pathology and our condition and function of our liver or associated organ.
These children were quite ill already.
Exactly. They have quite substantial pathology with the liver. And so in this case, then we have our gene therapy at a very high dose and a very, very high dose. Then we have collectively, we have toxicity from our viral capsid itself. We may have toxicity from our transgene product. We may have a toxicity from our innate response to AAV, both a capsid protein and vector genome. Collectively, all those may trigger further worsening of the liver function and cause very tragic death of the patient.
Okay.
Patients.
Not just this one, but there have been several instances in literature recently that I've seen that points out that the sheer number of vector can trigger an adverse event, a serious adverse event. And this implies that a more potent vectors need to be developed so you can use less of them. How are you thinking about doing that? And I think part of the answer here goes to your first stop on your road trip, which is manufacturing.
Yeah, so you're right. The one key challenge in manufacturing of AAV is in the manufacturing process. AAV often was made in 90% of viruses or variants, they are empty. They do not carry or gene therapeutic payload. And in that case, and only 10% are really going to be potent and effective vectors. So one of the major challenges for manufacturing people is to figure out a way to separate the empties from the functional vectors.
Yeah.
So by doing so, you will enhance the potency of the vector genome. You will reduce total imaging node, meaning that you have less viral particles and more functional viral particles. And by doing so, you may reduce toxicity. You may reduce the dose that as you expected.
If it's not too complicated, could you envision or explain to me how you can detect an empty virion? That sounds.
Yes, so they basically, a functional virus are two components. You have your therapeutic gene component inside of the virus. You also have the capsid component, to cover or carry those genes. However, when you have an empty virus, you only have capsid. So by determining the biological components of your gene therapy vectors, you can determine or use physical methods such as electronic microscope or such as ultracentrifugation and many other methods. You can differentiate that a virus that is empty or carry your therapeutic payload.
Okay. Actually, this isn't mentioned in any paper that I've seen so far. How much time does it take? If I want to say, I want to scale up this vector I made, I want to treat 10 patients. What kind of lead time do you need to do that?
This is a complicated issue because there's a whole supply chain or issue because current method to generate AAV got to start with plasmid DNA. So you have to generate GMP grade of vector plasmids and the packaging plasmid and the helper plasmid. And then you get into vector manufacturing stage. And then you had to do a lot of work to figure out or validate the cells you use. And then you perform the production process. And then you perform harvesting and the downstream processing and the purification process. And then you purify your vector. And then you characterize your vector through fully certified good laboratory practice. So all these things, it depends on whether you get a place who can have opening to do the production for you and the whole process you need to characterize it for you. So I would say there's a very difficult direct answer for this. It depends on the availability and the whole supply chain availability.
Okay, so this leads nicely into my next question. This is expensive. I mean, it's a lot of work, it's a lot of reagents, it's expensive. So I want to quote from your paper. Quote, although treating a monogenic disease by recombinant AAV vectors has now become more feasible than ever, the high risk and high cost of developing AAV-based investigational new drugs discourages many investigators from translating preclinical studies to clinical trials, end quote. And then when we spoke recently about your own work, you said to me, the only way to translate your concept into a therapeutic is to start a company, which you did twice. One's called ASPA and one's called Voyager. We'll talk about that a little bit later. But in general, are you concerned about the high cost of entry here is made as a business proposition, made it too difficult for a lot of people to think about?
I do. I have to say, as I said in my slides, the very first barrier facing commercialization and translation of gene therapy is the manufacturing. So at this point, the cost, because our manufacturing methods, our manufacturing process, our yields, potent gene therapy vector yield per cell limited us. And also our process of separate empty virus from functional potent vectors can limit us in terms of produce high quantity, high quality vector in a short amount of time with reasonable resources. So that is definitely going to be a limitation for us. But at the same time, I had to say, as academic researchers, and our focus is primarily our capabilities in early research development to generate preclinical proof of concept drugs However, in order to push this clinical translation, there are many important steps we have to do. In research, we can make vector as we so-called research grade, and which is relatively simple and cheap. But if you want to put into human, we require good manufacturing practice. That is really clinical vector production. Because of the quality control, quality insurance process that make this manufacturing much more expensive. So, and for most of academic researchers, this is not affordable. And I'm happy to see now NIH now start paying attention to start supporting the early translation for clinical trials. But the most effective step, I personally believe, is really link up with industry people, with pharmaceutical companies, with biotech companies that take advantage of their much more resources, their experience in drug development, their experience in regulatory science, their experience in biological manufacturing, and all those add together, and that will push gene therapy and all novel therapy move much faster.
Thank you for that. Let's go on to the therapeutic cargo. I mentioned this a few minutes ago. If we could circle back and put up the graphic again of the actual structure of the AAV. I want to talk about what you can actually do with it. And that is, how much of the original genome of the AAV vector can you lose and still use a therapeutic? There we go. So how much of this middle part can you lose and still have a viable vector?
Yes. So basically, this part is outside part. It's how AAV dresses. Actually, the diameter of this virus is 26 nanometer. And however, it has a very limited cargo size. In this case, maximum you can put in is your therapeutic gene, probably with all regulatory gene expression accessories together. It's about 4,600 base pair. That's maximum you can squeeze into this.
That's small.
And very small. Compared to COVID-19 you mentioned, this is nothing. So that is definitely a challenge for many diseases that with larger genes, such as Duchenne muscular dystrophy, which have cDNA of probably 11 kb, and such as cystic fibrosis, which has the genome of easily about 4.5 kb, it becomes very challenging. Cystic fibrosis gene.
So to contrast this, is something I'm familiar with, which is CAR T. And one introduces the genetic material to make a CAR T into a cell using the lentivirus, which can hold a much larger cargo. You could not do a CAR T with an AAV.
It's unlikely.
Okay.
Unlikely because lenti can hold up to 10 kb or 10,000 base pairs. And it's almost doubled of AAV.
So are there ways around the size limitations, some way to deliver a larger payload with the AAV?
Yes, people have been trying to develop this so-called expanded capacity AAV. And you can do that through 3 different mechanisms. One mechanism is you split your gene into 2 parts. And through different mechanisms, the vector genome can recombine, become a bigger genome inside of your cells and express a larger gene. The second mechanism is you also split into 2 vectors, generate 2 different messenger RNAs, but those messenger RNA can combine into a larger messenger RNA through trans-splicing mechanism. And then you can express a larger gene. The third method is so-called protein recombination. In this case, you express 2 different proteins from 2 different vector genome, and through an intern system, that protein can splice together, become a bigger protein, and for gene therapy. So those are the 3 methods people have been trying.
That's interesting. I've not heard of the last one. Would that be, are there therapeutics where there would be two subunits of a protein that you could then express together?
Correct. You
Okay.
could mechanism that they can trigger protein splicing and get them together.
Okay, but that means you have to get both vectors in the same cell. So,
correct. That's a very good question. That's a brilliant question. It's the precondition is doesn't matter you do two vector system or you do trans-splicing of the messenger RNA or splicing of the protein. No matter what, those two viruses got to get into the same cells to make this happen.
okay. Again, more challenges. Now, the next thing to consider is destination number 3 on your Highway 66, and that is tropism, meaning you don't want this genetic modification to land anywhere. You generally want it in a specific tissue type. So the issues here include choosing the AAV you want for your given tissue. Now, granted, we're going to put up a graphic here. This is a pie chart, which is this is all the places we're targeting and all the ways we're trying to target this. Granted, this is from your 2019 paper, so the number of clinical trials has certainly changed since then, but let's look at the tissue. Those most popular vector destinations are the eye and the brain. And I know this makes sense because those are, quote, immunoprivileged spaces, so they're a little safer for your vectors to survive. So that makes sense. But the other ones we see here are liver and muscle. So what makes those tissues more amenable to gene therapy than, say, the heart muscle, which has the smallest slice of this pie?
Yes, I think it's one of the major thing is, again, the delivery. I have to say, to target particularly muscle, liver, and heart, they all requires systemic delivery, meaning you do IV dosing of the gene therapy vectors. And once you do IV dosing, then the tissue tropism and specificity become important. You want the vector ideally to only target the tissue of your interest, like you mentioned, heart. However, when you put virus into the bloodstream, many different viruses automatically go to liver. That become a depot for many viruses get there. And then that is one of the reasons for this Alden's trial is that our targeted tissue may not be the liver, but the virus inadvertently going to get into the liver and cause your toxicity. So that is why the ideal AAV should be only target the targeted tissue of interest, but not other tissues. That is a major challenge. And also that will significantly reduce the toxicity if you could accomplish that.
If you had, I don't know if this is a good question or a smart question or not. If you had an AAV that naturally targeted muscle, but you really wanted to target some other organ, but the AAV that does the muscle is actually really stable, so you'd like to use that one. Can you mutate that to have a different tropism, or do you have to go with a different vector altogether?
That is really the direction people have taken. That is, evolve the vector in a way in this targeted tissue and to generate the targeted tissue specific virus or capsid to accomplish that. So that is definitely one of the major directions for the field to achieve that.
Okay.
By evolution.
So now the final stop in your road trip is gene expression, where you wrote more is better is a bad idea. So using the example of a disease where you lack something or you don't have enough of something, once you have a stable integration of your gene, you need to control the expression of that therapeutic protein, one would assume. How are you thinking about doing that? I know how to do it with a CAR T, but this is a much different beast.
Yeah. So the ideally, you want to gene expression at natural level, meaning that your body regulate your cells regulate its expression level. But that is hard because a lot of the genes, we still do not have a fully understanding how natural genes regulate itself. That's understand the mechanism. The second limitation is when you use AAV, you have very limited transgene space. So in that case, promoter will count your capacity. A lot of natural promoters, they're big because they have a positive regulatory enhancer, negative regulatory enhancer in responding to your physiological needs. So to accomplish cell type specific physiological level of expression, you may have to put a big chunk of the promoter to achieve that. That will limit your transgene, your therapeutic gene size, and that become a challenge, to this. Alternatively, you could use small molecule drug to regulate your gene therapy. That's called the pharmacological regulation. But the problem with that is people have to figure out a way not only express your gene, but you have to express your controller, meaning that that controller or operator, it determines your gene expression, and that operator alternatively should be controlled by small molecule. You give the small molecule that operator turns on, and then your gene turns on. And that is, again, the ideal level. When you need it, you express it. When you do not need it, you keep the silence. And that is physical by science, but it's very difficult to be practically accomplish that in human patient yet. But that's definitely an area we should work towards.
Yeah, I'm actually very familiar with the last approach from the CAR Ts, where you can actually put in a switch within that whole construct. And then the switch comes on the surface of the cell, and you hit that with a small molecule. You can even titrate the switch.
Correct.
But yeah, this is the different.
Same principle, but different applications.
So now I want to talk about what one might want to do with the target tissue. And this goes to the genetic problem that the patient is having and how you might fix it. There are 4 potential strategies that I want to mention here. The first is gene replacement. This strategy aims to deliver a gene product to compensate for the loss of function mutations. An example here is the genetic blindness that is cured by a drug called Luxturna, which was approved quite recently, a very remarkable drug from Spark Therapeutics. The second example would be gene silencing. In contrast to the replacement, gene silencing mainly tackles monogenic diseases caused by gain of toxicity as opposed to gain of function. Huntington's disease is an example of this, and the way you might want to fix that is with RNAi, which you could insert, which then dampens the gene expression that is causing the toxicity. Another one, gene addition. This is interesting because one of our clients here at LifeSci, a company called Geneta, is doing this. A gene addition strategy could use the AAV to deliver genes encoding a drug, actually. And Geneta is doing this with glioblastoma, and they're delivering interferon gamma as a therapeutic. Then finally, gene editing. This therapeutic gene editing typically occurs in 2 steps. You have a generation of a targeted DNA break, and then you fill it in with something better. Everyone's talking about this, it's CRISPR. So Dr. Gao, of these, what has been the most successful and what has been the most challenging?
Yeah, the most successful is the #1 concept, that's gene replacement. This is a traditional definition of the gene therapy. When you have a malfunction genes or not defective genes, you just replace it with a functional gene. So that many gene therapy successes includes the Sparks externa, Luterna, as well as the Vexus drug, the Gensma, they all belong to this category. Yeah, that's the most successful, or we have more experience with. The challenging ones would be those ones we have less experience or understanding, includes the gene silencing you are talking about, as well as gene editing. And those are the ones that are more challenging because the concept come up more recently, and we have less human experience with them.
Well, I want to bring up the example of the gene editing modality because this was just in the news. The company called Intellia, that works with CRISPR, and on June 28th, they announced that they had the first positive data for in vivo gene editing with CRISPR for a condition called amyloidosis. As one of the leading researchers in the world, Dr. Gao, was this a big deal?
This is a huge deal. This is the first demonstration that you can apply this novel approach in human body and to accomplish your goal to treat a human genetic disease. And again, this is a big, big deal. This is the first proof of concept that gene therapy can use in vivo, in human, and editing and treat the disease.
Okay. Now we're 12 minutes on to the hour. We're into the Q&A portion. For those of you listening and you have a question, please, any question is in bounds. I know this is a rather complicated topic, but go ahead and dial in now. And to service the investor on the call, I have a question for you, Dr. Gao. So now that we have this foundation, how about some advice for the investor who's not familiar with this space, but he needs to be able to take a quick read on this new investor deck that just came across his desk? If you could, for me, describe to me, when you open up a platform and you look at things, describe to me a short list of things that you have to see in this platform for you to know that's going to work.
Yeah, I lost the part of your conversation because of the internet issue. I'm sorry. I missed the important part. Could you repeat, please?
Yes. So for the investor who is not a geneticist, could you give us a short list of qualities that you need to see in a gene therapy platform to know that these people know what they're doing?
I see. So before I get into the question, I want to make one correction about this gene editing reported by Intellia. Actually, this is one of the first example of demonstrated gene therapy works, gene editing works in human. There's some other products in that category. And about your specific question, I have to say, if I am a reviewer, if I'm interested in considering investing this company or this product, I first have to look at efficacy, how effective it is. And second, I look at the safety and whether they have any side effects by this treatment. And third, I need to look at manufacturability. Is this how easy the drug can be made, and what's the cost of it? And fourth, I will look into from a financial point of view, from a research point of view, probably we pay less attention, but financially we need to look at a market, like patient population and other things.
Okay.
And those are things I will look into.
So now we've come full circle. We've got 9 minutes left. And since I'm the host, I can ask anything I want. So I'm going to ask about Canavans disease. Tell me the story of the day you discovered the Canavans gene.
Yeah. So Canavan disease is a very important in my scientific career. I started joined scientific research 1989 at Miami Children's Research Institute. And that's the time I get into rare genetic disease research. I spent 4 years, 4 years in that 5 years, 4 or 5 years in that institute. But what it was very impressive to me, where I always think about it, is like this day. The day right after Hurricane Andrew, that I don't remember which year, and after the overnight big hurricane wiped through Florida. And
you were in Miami.
Oh, I was in Miami, and then I said I have important tissue culture going on in the lab. No matter what happened, I got to make the lab to see that, because that it would be the first time we know we can produce, our gene can produce the product that can treat or potentially treat the disease or diagnose the disease. So I drove out everywhere, all the traffic lights and everything was broken down. You can see cars here, there, and big trees on the roads. But I was able to, manage to drive about probably 12 miles, get into Miami Children's Hospital eventually, and I open up my culture and start doing assay to see whether I generate the enzyme called aspartate, which is what we're missing with Canavans disease. And then I did the test with a colleague of mine. And when I saw those color changes and also our reads like shoot up high, we say, we got it. Now we have the gene we can use for diagnosis and also for therapy. So that is I will never remember that day, that morning, what happens in my scientific research career.
So a terrible day for Miami, but a great day for you and Canavans disease patients.
Yes.
There is also a company here that Dr. Gao formed called ASPA to treat this disease, but we're not going to talk about more right now because we do have a question in the queue. Morgen?
Yeah, and thanks, Dr. Gao, for that story. I love that. It's a great image, imagining you making that discovery with the crazy weather outside. So one of our listeners has a question for you. When do you anticipate that gene therapy will be ready for treating common or chronic diseases?
Yeah. Morgen, that's a great question. Actually, the gene therapy, what I have seen, it's gradually migrating. We start with first and with clear pathological cause or gene defect disease, we call ultra rare disease, that we know exactly what's the issue. And then we're gradually migrating into rare and moderate rare diseases. And now I see there are companies in the field that started working on the disease, some more general population chronic disease, such as diabetes and such as other metabolic diseases. So I do see this migration is happening, and it's happening fast. So I would say, think about the gene therapy, real gene therapy efforts really started back in 1990s. And that's the first clinical success in human patients. And that use retrovirus similar to lentivirus for ex vivo gene therapy. And then back to, like early days, early 2000s. And then we have some initial success with AAV in vivo gene therapy. So that's already 1 decade past. So now when we have the first 2 drugs of AAV get approved back in 2017 or 2019, those drugs get approved. So now, how many more years we can have the gene therapy drug to treat chronic diseases? I would say my view is probably within 5 years, we may have some drug at the very late stage or early stage of clinical evaluation.
Wow.
Morgen, while we wait for more questions to come in, I wanted to chime in with another one. Dr. Gao, I know you are the champion of AAV, but some people talk about non-viral vectors. What do you think about those?
Yes, that's a very attractive delivery platform. Actually, I have to say, viral gene delivery and the non-viral gene delivery have been really developed in parallel in the past decades. I still remember when I entered the gene therapy field, 1994, that time, a lot of research being done, not only on virus, but on non-viral delivery. And recent examples of some success of the approved drugs from Alden and clearly demonstrate non-viral gene delivery is workable and clinically translatable for treating human disease in vivo directly. And so what I had to say, and also most recent success with messenger RNA vaccine, I mean, that is really delivered. This is for COVID-19 and other applications of vaccine, molecular vaccine, genetic vaccine. Those are really depends on non-viral platform to deliver messenger RNA into our body to trigger immune responses. And so from that point of view, I still think non-viral is a very important part of a gene therapy delivery platforms. And most importantly, for gene editing, probably non-viral really holds the hope. And because the CRISPR, no matter what, it's bacterial protein. If we use a viral gene therapy to deliver it, we may generate some sustained immunological responses problem. But non-viral with a transient delivery, particularly with a messenger RNA delivery, we may have less immunological responses. So, and from that point of view, I had to say non-viral is a very important part of a gene delivery platform technology for gene therapy.
Okay, we have just a minute left. Since we started a little bit late, if you have a question, please go ahead and squeeze it in. I can go ahead and make room for that. But if not, I'm going to go ahead and ask just one more of my own. Dr. Gao, you founded another company. It's called Voyager. And I won't ask you about company operations. You have nothing to do with that. But as for the science, Voyager is focused on Huntington's, ALS, and something called Friedreich's ataxia. Why did you choose these diseases to work on?
Yes. So first of all, I have to share my views on Voyager. Different from ASPA Therapeutics, Voyager, I more consider it as a platform technology company with a focus on CNS diseases and with gene therapy to CNS. So that's more recently, as you may know, Voyager developed some gene therapy vectors that is a very highly tropic, can cross blood-brain barrier, efficiently target CNS. And second, about the disease selection, I think Voyager from early days, their target selection for gene therapy, it's more focused on moderate rare or less rare diseases, such as Huntington disease. Unlike a Canavan, which is ultra rare disease, and Huntington is more has a broad, higher prevalence disease. So they are indeed focused on Huntington as one of their drug development targets.
Okay, Dr. Gao, Morgen, thank you both for joining us today. Thank you all people at your homes, your desks, your beach houses, wherever you are. Thank you for dialing in. Morgen, please take us out.
Yes, thank you. Thank you, Dr. Gao. Thank you, Neil. Thanks to all those who listened in. Have a great afternoon.
Bye-bye, everyone.
Bye.
Thank you.